I'm going to give an overview of some of the opportunities and also some of the issues that you need to consider as a new model of blood banks moves forward, the new business model and new approaches. As Sean mentioned, doing business the old way simply is not sustainable in the long term. We've seen a steady and consistent decrease in diminution in revenues and sustainability of blood banks over the past several years, and there's no obvious reason to believe that won't continue. So modifying your business model at the blood bank to maintain sustainability, we are after all .orgs, is going to be key to our survival. And also, it's an exciting opportunity for all of us because stem cell research and regenerative medicine, we're at a nexus right now where there's going to be breakthroughs in medicine and a high demand and expectation from the community at large for better therapies, but also an interest in them by biopharma and developing them. And we're at the cusp of that, and we're at an opportunistic place. There's also a lot of pitfalls to consider, though. As I mentioned, cell-based therapeutics are widely expanding area of development for regenerative medicine, transplantation, oncology, and immunology, and other indications. Inherent in that is the GMP manufacture of cell therapies, and this requires cell sources that can be utilized for fetal layers, cytokine sources, and cell product progenitors. And I'm speaking specifically to our personal experiences currently at the San Diego Blood Bank. We actually are involved with researchers and biopharma entities doing these types of activities, both at the research level as well as in clinical trial enablement. These cell sources, a key point, they must be well characterized and produced with rigorous manufacturing and control guidelines. The agency's always said potency and identity. We need that. Even with fetal layers, there's some impact there. You have to be able to identify what you're doing. CGMP qualifications, accreditations such as CLIA, AABB, or FACT, and FDA compliance must be met. And blood banks are well positioned for these because of the products we've generated over the decades, have these oversights in place already. This is just for cord tissue therapeutics. This is just a graph showing the explosion, how from 2004 to 2012, there's been almost an exponential increase in the numbers of clinical trials simply with mesenchymal stem cell-based therapeutics. The same thing is holding for CD34 cells and other lineage-committed progenitors that are being used as therapeutic approaches. So it's a remarkable growth industry area and opportunity for all of us to be involved with. One of my predictions is that right now CD34 cells are a great focus for us and a great need, but mesenchymal stem cells are growing and may eventually become as important or maybe even overrun as soon as clinical trials prove that they work, a demand in terms of biopharma or from a therapeutic standpoint. Here's some of the potential products and services a blood bank could offer. Obviously, PBMCs, mononuclear cells, and red blood cells that can serve as fetal layers or for cytokine production can be used, and we've actually used some of these, and we do just that for companies that are in clinical trials. You can also be participating in assay and device development, sometimes supplying cells to validate devices or to set up assay. Also, obviously, serum, platelet, and plasma components are a growing area of interest. The agencies, both EU and US, have been guiding us to get away from using fetal bovine serum. It's difficult to get. It's easier to get human platelets than it is to get fetal calf serum. So using platelet lysate products or platelet-derived products as an alternate media supplement to grow your human cells that you want to use to treat patients within the clinic is a favored approach right now. Ultimately, purely totally defined media that's serum-free and has no animal components is where we'd like to get, but right now there's still a deficit in terms of those defined media always working. And it looks like things like platelet lysate products show great promise. And there are a number of companies actively pursuing this area. Obviously, cord blood and tissue cells for transplantation, lineage-committed progenitors, T and NK cell-based therapies. We've all heard of CAR T cells. Well, now there's CAR NK cells. And you might be able to use this as a source material. T cells may be a bit more difficult, but NK cells, there's been a number of robust papers now showing you can readily make NK cells and NK progenitors from cord blood progenitors. So there may be an opportunity there as well. Mesenchymal stem cells, duh. I mean, it's on everybody's lips right now at all the meetings I go to, and clearly it has great promise, but there's also, picking the right type of mesenchymal stem cell is going to be important. A lot of the trials right now are autologous, where you take a person with an autoimmune disorder, and inherent in that is the inference that the mesenchymal stem cell milieu of that patient is somewhat flawed. Well, they're taking those MSCs out of that same patient, making more of them and putting them back in with the expectation that more is always better. Maybe it will be, or maybe we need to do a better job of, once again, potency and identity, identifying the right cells that we use. Cord blood banks and blood banks in general have an opportunity to be actively engaged in that by supplying healthy donor mesenchymal stem cells for characterization and setting benchmarks. And the new and growing area is the precision medicine aspect of this, bioinformatics. Blood banks are uniquely positioned to longitudinally evaluate tens of thousands of normal, healthy donors over their lifespan. We have donors that have been coming into the San Diego Blood Bank for 20 to 30 years, 100-gallon donors. And every time they come in, we get a sample of their blood. If we can now put in place IRB clearances and consents to allow their blood, their genome to be analyzed over time and track how a person goes from being healthy to unhealthy and what gene signatures shift, especially now that you can power it with this large number of people, you may have a much more precise way of defining those genetic elements or epigenetic elements that are key to disease or key to better treatments.
That's brilliant. That's brilliant.
Key points to consider when you access blood banks, though. Check their resources and infrastructure. Make sure we've got the bandwidth to help you out if you're a biopharma that's looking to work with us. Make sure they have a robust quality and regulatory oversight team. We certainly do. Helen Bixaman is my best friend in the world. She wasn't till I moved here a year ago, but she's my Vice President of Regulatory Affairs and keeps us out of the weeds and keeps us on track. Source material. We do things like donor recruitment, consent, IRB clearances, IDM testing and tracking, and other types of assays on behalf of our customers because we do that anyway for the patients that we serve. So some of the advantages are blood banks have well-established CGMP compliance, accreditations, quality and regulatory oversight for cell products being produced. We have a large recruitment and donor base for collection of source material. And we've now matured in terms of our consenting strategy to make sure we're assured that we have all the clearances from an IRB standpoint and from a consent standpoint for use for research purposes. You have to be very careful there. A lot of people have naively said, well, if you consent to research, it's okay, you can use it for anything. Well, no, it's a bit more subtle than that. You have privacy rights you have to address, commercialization rights you have to address, and you need to make sure you're very transparent to the donor about this. And we've done that. We've gone to great lengths to make sure no one is not aware of what's going to happen with their material if they agree to allow it to be used for research purposes. You also have established infrastructure and trained qualified personnel for production of products. We go through periodic retraining and competency across the spectrum of all the activities we do at blood banks. So it's a powerful opportunity for biopharma and others to work with us. We're already trained, we're already qualified, we already have the oversights from a safety standpoint that we need to generate products for people to use. We can serve as a single site for consent, processing, testing, sterility, irradiation, isolation, enrichment, and process storage and distribution. So we can be your one-stop shop for a lot of this as well. In conclusion, we're well positioned to collaborate with industry and academics in the realm of cell therapies. Complex laboratory and regulatory issues can be managed by blood banks. We do that on a daily basis anyway. But we've been through 4 or 5 audits in the past 8 months, and successfully so. Part of that is when you deal with a customer is what is known as a CGMP audit, customer audit, to make sure you're CGMP compliant for their product that they want to make. So we have a new era of stem cell-based medicine. It's rapidly growing. Focus, diligence, quality assurance, regulatory guidance, consent are all key to generating products that can be used to help to make this world a better place. Thank you.
Rob very deftly handled the idea of opening up this notion of how what services groups like us can offer. And back last year, I don't know if anybody was here last year, we had a session on how advanced blood banks can help bridge the gap, the kind of the valleys of death that we've all heard about between a scientific advance and a medical breakthrough. So last year it was all about what we can do. This year what we hope to do is talk about what we are doing. Okay, and so some of the things that Rob was talking about, providing feeder layers and things like that, those are things that they're already doing, they just can't talk about. I'm going to talk about a little more things about what we're actually doing because we've gotten the approval to do so. So first of all, who is BioBridge Global? So BioBridge Global is the parent organization of 4 501c3s. So it's a 501c3 nonprofit holding company and 4 501c3 functioning subsidiaries. And the emphasis there is on functioning and operating subsidiaries. So really the first 3, Qualtex, South Texas Blood and Tissue, and GenCure are the operating subsidiaries. The 4th is a foundation. So Qualtex is the largest independent not-for-profit blood and tissue safety testing laboratory in the United States. We test on average about somewhere around the 8 million samples mark, probably about 40,000 samples a day, and a vial of blood is a sample. So 40,000 a day. It's testing on an industrial scale, and so we're moving trying to think of what else can we test. All right, so cell and cell-based therapeutics are an area that we're considering as well. So that's one thing. I'm not going to really touch on that much. South Texas Blood and Tissue Center, that's the blood bank, and once again, industrial scale blood bank with network partners. We have platelet lysate. We sell outdated platelets and just as everyone else does. But and then GenCure, that's our not-for-profit blood and tissue safety, really regenerative medicine group, and I'll go into more detail. So company statistics, 750 plus employees, that's old. We have locations both in Texas, San Antonio, and Norcross. Just north of here, Atlanta. So go figure. Last year, the World Stem Cell Summit was in San Antonio. Here it's in Atlanta. Go figure. It's great. So projected revenues, about $230 million in 2018. So we're a not-for-profit, okay, but a pretty darn big not-for-profit, okay? So, and we live and breathe in the not-for-profit world. People talk about, blood banks, you're a not-for-profit, you don't understand. Okay, it's excuse the pun, it's a blood sport, it's a blood bath. I mean, there's a great deal of price competition in this area. So get that whole not-for-profit, everything comes easily to them, out of your mind. So we really do live in the for-profit world. And the real operations world, let me put it that way. The biggest growth opportunity that we've seen is in the area of regenerative medicine and cell-based therapeutics and products. And these are offerings for which GenCure, and I'm the interim chief operating officer of GenCure, we will play, I think, a leading role. So who is GenCure? As I said, we are the regenerative medicine's focused subsidiary. We have 3 groups. One is the GenCure Tissue Center, then there's the GenCure Cord Blood Center, and GenCure Cellular Therapy Center. So the GenCure Tissue Center, I'm kind of speeding through these. We're essentially, GenCure is an orthobiologics company right now. That's primarily where we get most of our throughput, if you will. But we provide human tissue allograft implants for hospitals and clinics for use in orthopedic replacement procedures. Spare parts. You blow out an ACL, you kind of need a new one, all right? So we're a good friend. We're the only, GenCure Tissue Center is the only full-service tissue bank in Texas. And by full service, that means that we can recover, we can process or manufacture and distribute. So we're vertically integrated that way. We've really tried to think hard about those sorts of things. Again, the only one in Texas, and Texas is a big state, AATB and ISO approved. We have the GenCure Cord Blood Center, jointly funded by the Texas legislature. So people talk about, gee, does it take an act of Congress to work with you guys? Well, no, but it did take an act of Congress to set us up. So again, we're the official cord blood bank of the state of Texas. We obviously collect and process umbilical cord and related tissues for clinical use, and we'll get into that in a little bit, and research partner with firms, biotechnology organizations, looking about how these things can be used. And then lastly, the GenCure Cellular Therapy Center, a home of Be the Match, the National Merit Donor Program, which you guys are all familiar with. We collect peripheral blood stem cells for hematopoietic transplants. We're unusual in that we have about 80-85% minorities. So we're fairly in demand, not only on the PBSC side, but also on the cord blood side as well. And we participate in 1 FDA-approved therapy and 4 clinical research studies right now. So we're doing both, clinical, clinical use, clinical research, and basic research all in one shot. So what do we want to do? All right, BioBridge Global. This was actually an ad for from the World Stem Cell Summit a couple of years ago. Our goal is to help enable regenerative medicine. That's really what we want to do, and we're already doing it. This is somewhat old, so let me show you what we're already doing. All right, so this was a press release that went out a couple of days ago where StemBiosis and BioBridge Global are in a collaboration to provide human umbilical cord blood cells for research use. And notice I said cord blood cells. What we want to do is, if you're offering something that everybody else does, we already do that on the blood side. All right, blood is blood. What we wanted to do was offer a type of mesenchymal stromal cell or stem cell that's unusual, all right? So anybody will tell you that the number of true mesenchymal stromal cells in umbilical cord blood is very, very small, all right? Well, this company, StemBiosis, has come up with a way to isolate many more umbilical cord blood mesenchymal stromal cells using their product called their HPME, or high performance microenvironment. Basically, the brilliance of this is they didn't try to outthink nature. Okay, so the bone marrow niche in your bones, they build their own house. They make their own structure. They build their own house. And so Dr. Xiaodao Cheng, who founded the company, thought, well, let's not try to outthink it, all right? So he took out bone marrow aspirate, you put it on, you plate it, and cause the bone marrow stromal cells to lay down a matrix of an extracellular matrix, basically build their house. Remove the cells, and then you have a substrate then that is perfect for keeping cells in their phenotype. All right, so they offer this, as you see at the bottom, as a research product, and we had opened our doors to them a couple of years ago to build out. So remember, we're a not-for-profit, but we invited them in because we're really trying to get closer to the biotechnology world. And so they are literally under our roof. They have their own little sovereign territory, and that's where they make this GMP grade material for expanding cells. So pretty brilliant stuff. But beyond that, remember, so we have the Texas Cord Blood Bank. We have access to lots and lots and lots of umbilical cord blood. And so what we decided to do was take these cells that StemBiosis isolated, expand them, actually pull out the colonies, expand them on our HPME to the level, to the number that then we can take and then put into the Terumo BCT quantum cell expansion system. And I see my colleagues from Terumo BCT over there, and thank you very much for coming. So it made a great deal of sense to me and us at the time, because remember, except for maybe adipose-derived mesenchymal stromal cells, any cell that's going to be used, any MSC that's going to be used, will have to be expanded in some form or fashion, okay? Because they just don't exist in that many numbers until you expand them to be used clinically, okay? So what occurred to me, well, gee, let's go with a system. Let's build our real options bets in these areas they're kind of set for scalability. All right, so the notion here was, let's take these umbilical cord blood cells that exist in very, very small numbers, expand them on their HPME, keep their phenotype, stick them in this machine, expand them in great numbers, and then offer them as a research use only product that is quite rare. And so that was the idea here. And we expect that our friends from STEM Biosys will be selling these probably in the somewhere in the July timeframe, something like that. So very excited about that. And then next, so something that we're already doing or have been doing is working with a company, X Biotech, out of Austin. They have come up with a thing that they call the true human antibody discovery process, where basically they take B cells from our donors, scan them for a certain type of antibody that they're looking for. Once they find them, we call them in again, get their live cells, they go into their genome, get the genes responsible for making that antibody, and then it's a true human. Everybody talks about humanized, it's really close, it's murine and human. No, this is true human, it's really, really human, all human antibodies. And they're in clinical trials right now for MRSA, and IL-6, an antibody against IL-6. Both of those came from donors that we provided to them, and they're doing quite well, and I'm very, very excited. And if you'll notice there, this picture at the bottom, you noticed Ebola. Right now, Ebola is, it's kind of passé, but remember about this time last year, everybody was quite freaked out about Ebola. Partly because this person, Amber Vinson, the nurse at Texas Presbyterian Hospital, got Ebola. We brought her in as a donor, and by definition, if you've survived Ebola, you're making the antibodies. All right, so we pulled out her blood, gave it to X Biotech, and they're going to be going into clinical trials for an anti-Ebola therapeutic antibody. New GenCure initiatives, I'm just about done. We just made, and this is kind of not new to the world, but new to us, a proprietary demineralized bone matrix product. Remember, we're an orthobiologics company, so we're a not-for-profit, but we're moving in the area of providing these value-added services on the materials that we already provide. And we will soon offer therapeutic apheresis services to a large local hospital. I just can't talk about which, but it's a large local hospital. So again, what we do, and that's a picture of Vanessa working with a patient to pull out her peripheral blood stem cells, that's what we do very well. So let's rely on what we already do very well and offer these additional value-added services to others. And then I think finally, we are sourcing amnion tissue for wound care companies right now, and that's a picture of one of our techs holding up what I call goop in a bag. So that is a placenta in a bag, and excuse me, that's one of the first photos. Be careful what you let me take a picture of, because it'll often end up on a slide. But it looks much, much better than that. But we will soon, in addition to just offering goop in a bag to these product companies, we will soon manufacture our own amnion-based wound cover product based on the products that we already have. And the idea here is, look, we're not trying to take over the world, okay? It's all about being sustainable, okay? what we intend to do is take these products that either we never bring in the door, or that we bring in the door and end up as medical waste. Take those products, add value, and then take those resources and plow them back into the organization so that we can do more good, all right? Do well by doing good. That's the plan. All right, so enough of me. Next up is Jeff Davis, who's going to talk about his things with Angiocrine.
Okay, thank you, Sean, for inviting me to participate. We are a small biotech company working in the field of regenerative medicine. We are part of this group here, not because we've committed to working with the blood centers, but because we are working on a program that clearly is very relevant to the blood centers, and we obviously understand the need to work with the blood centers and understand what they have to offer. We've been having a lot of conversations and understand their capabilities. It's been elucidated by Rob very nicely already, and really understand we need to work with them, but still trying to figure out just how. And by the same token, we believe that we have a program that's going to really contribute to the blood centers as well. So it's going to be a symbiotic relationship ultimately once we figure it out. So we are the E-Cell company. We are committed to regenerative medicine. I'm going to tell you about one program today, which is a program in cord blood stem cell expansion. The company is based on science from the laboratory of Dr. Shaheen Rafi at Weill Cornell Medical Center. And Shaheen's research has been based on the understanding that endothelial cells are something more than just conduits. And in fact, that they really provide an executive function to stem cells and more or less tell stem cells what to do. And they do this through cell surface molecules as well as through secreted factors that Shaheen had dubbed angiocrines, a term that now is very common in the lexicon. In order to really translate that scientific wealth of information into something that has potential for therapy, Shaheen made really a serendipitous discovery that he could introduce a single gene, and this was a gene called adenovirus E4-ORF1, into endothelial cells. And this would endow endothelial cells with the ability to proliferate, not quite indefinitely, they're not immortalized, but to undergo many serial passages so you could expand these cells into numbers relevant for therapy. And importantly, it also endows these cells with the ability to be expanded in the absence of serum. So we talked earlier about the importance of getting away from serum. With these cells, in fact, you can get away from serum. They will grow in serum-free media. And what's nice about that is that then enables the proliferation of stem cells that retain their stemness in culture. So we can use these cells to expand stem cells that retain their stem-like property because we're not introducing serum with that gamish of exogenous factors that can send stem cells every which way. We've recently succeeded in actually moving this system out of the flask and into a closed system. I can't tell you the name of this device because we have an upcoming press release, but those of you with good short-term memories can probably figure it out. So we've been able to use this system. We're working with Gerhard Bauer at UC Davis to use the system in GMP mode to expand the endothelial cells themselves, but as you'll see, also to expand additional products. And so this is a totally automated system, a closed system, very portable, and gives us a lot of options when we think about downstream manufacturing. So using this system, we're actually thinking about three different kinds of programs, and have three different kinds of programs, well advanced. So on the right side, you'll see really sort of our lead focus, which is using the endothelial cells themselves as the therapeutic. And we have a number of publications indicating that these endothelial cells will foster organ regeneration in a tissue-specific manner. But the left side is the one that we'll be really focused on today, which is using the endothelial cells to expand stem cells for hematopoietic stem cell transplant. So why do we want to expand stem cells from cord blood? As I'm sure many of you are aware, cord blood does offer a lot of advantages compared to the other sources of stem cell transplant. Cord blood cells are inherently more plastic, and so there's a lower incidence of GVHD, there's lower rates of relapse. Cord blood is readily available. We have 600,000 units stored around the world, and so there's enough diversity out there to really be able to serve every patient who needs a transplant. But there are also a number of drawbacks to cord blood. And those drawbacks include slower rates of engraftment, higher rate of graft failure, but these drawbacks all stem back to one single issue, and that is just that your typical cord does not have enough stem cells in order to reconstitute an adult. And so the solution really seems fairly obvious. We're not the only people who have realized the need for this, and that is to be able to expand the cord blood stem cells. So there are a number of other companies and academics working on this. We think our approach is a little bit different, and we are using a cell-based approach. We think that expanding these cord blood stem cells is a complex problem and requires a complex mix of factors, but these factors can all be supplied by the same cells that supply the factors in vivo, which are the endothelial cells. So this slide shows the typical pattern of differentiation of stem cells, but really what you're trying to accomplish when you expand stem cells ex vivo is to keep them in their stem-like state so that when they go into the body, then they can undergo the differentiation. And again, using our system in this closed device, we can use the E-cell technology in the absence of serum to expand the stem cells to keep their stemness so that when they go into the body, they can fully reconstitute all the different lineages. We made these first observations, it's actually prior to the formation of the company by Dr. Jason Butler, who was working in Shaheen Rafi's lab at the time, in a 2012 publication. And what this slide shows is that using the E-cell technology, you can expand very primitive stem cells that will repopulate skid mice, both in a primary and a secondary fashion. As a serial transplant, they'll still repopulate succeeding generations of mice, indicating that these really are cells that retain their true stemness. And so based on this, we're looking at a product that is going to be derived from co-culture of the E-cells, and these are E-cells derived from umbilical cord, and E-cells with a matched cord blood unit. We'll rapidly expand these, and we can expand them in a clinically relevant time span of 6 to 10 days and expand 60 to 100-fold over that period of time. The stem cells and expanded cord blood cells will then be co-infused together as a single therapeutic product. And we believe that the endothelial cells are in fact playing a role in facilitating engraftment of the stem cells. So final product manufacturing, we're not quite resolved on how we're going to do this yet, but using the closed system, we have the opportunity of actually expanding the cells at the point of care or at select regional facilities. And of course, amongst those select regional facilities, the cord blood banks with all their expertise and their experience in regulatory and quality issues certainly comes to mind in its forefront. We now have positive preclinical proof of concept since that 2012 publication. So we've done a lot of work now, not only mice, but also non-human primates, with 2 academic investigators who are forefront in the field of studying stem cell differentiation in primates. And they've used our technology to expand stem cells that are barcoded and been able to determine that, in fact, these expanded stem cells will repopulate all the different lineages, both short-term and long-term. And we now have monkeys out that are over 2 years reconstituted with stem cells expanded from our technology. So we feel that this program, ultimately, is successful, is going to provide value across the full healthcare spectrum. Obviously, it's going to provide value to the patients in allowing them a safer graft with a lower incidence of GVHD, with both short and long-term engraftment. With the short-term engraftment, it should get them out of the hospital quicker and reduce the transplant-related mortality associated with now the longer delayed engraftment associated with cord blood transplant. But I want to also focus on the upside for the cord bank, where really giving the cord blood banks a better opportunity to provide a match allows them now to make better use of all the units that they harvest. Will be able to greatly reduce the threshold for acceptability based on the TNC count, and hopefully really open up the cord blood business and allow all those units that are currently sitting in the bank that are not useful to be useful. And in our wildest dreams, if this really works, I think that it opens up the door for stem cell transplant as a reasonable upfront procedure outside of the blood cancers where it's so prevalent now. And with that, I'll close and pass the baton to the next speaker. Thanks.
So welcome to Memorial Blood Centers, which merged with the Blood Center in Lincoln, Nebraska. If you merge a gopher with a husker, you better be innovative, hence the name Innovative Blood Resources. We do a lot of stuff. We are more fun than the average blood center. We recruit donors, process exactly all the stuff you've heard by my colleagues. We collect and process hematopoietic progenitors from patients, and for the National Marrow Donor Center, one of the testing centers for the National Marrow Donor Center. We actually are the home of the National Marrow Donor Center in Minneapolis, and so I am on their Human Subjects Committee. Our head of marketing was over talking to the Minnesota Twins and looking out the window and saying, oh, what's that construction across from the Twin Stadium? Oh, I think it's a dating company, the head of communication of the Twins said. Why is that? Well, it says, be the match. So before you come up with an innovative name for your company, make sure they know what it is. Just saying. We do testing for, like friends at Qualtex, donors, National Marrow Donor Program, fertility clinics. We test for over 130 fertility clinics. We test for the Iowa Mother's Milk Bank, which was a fascinating place to visit and a wonderful group. We do many clinical trials, which involves donor screening, collecting various products, as you've heard from my colleagues, but also providing a lot of products and components for trials. We are sort of the source of medical alley. There's a lot of medical device companies, so Medtronic, Boston Scientific, before they release a machine that processes blood. Yes, they run a lot of cow blood through it, but they also often need to do human blood. At the American Association of Blood Banks was this wonderful Korean company, NanoEntech, that had these amazing electrical engineers that kept showing their product each year. And I said, well, you know, when are we going to be able to use this to count the residual leukocytes when we take the white cells out of blood? And so we did not one, not two, but 3 clinical trials with them. And finally, I made them sit down with my FDA advisor, Sharon Orton, who's 10 years of experience at the FDA. As you heard this morning from the FDA officials, sit down with the FDA first before you do the clinical trials so that you are providing them the data they need. Anyway, we did that, and so this is a cute little device that is much cheaper, quicker than doing fluorescence-activated cell sorting. And so I said, okay, what about CD34? So we are now working with them, and they are developing this machine to count CD34 cells, which again would be very convenient for those of us that don't want to have to use fax for every CD34 count. I did want to, since my colleagues have done such a wonderful description of all the myriad groovy things that blood centers can do, to do a little deeper dive. And so my friend Al Dietz down at Mayo was kind enough to share some slides with me from their development. Mill Creek, which is basically making platelet lysate like the San Diego Blood Center. And so you've heard a lot about mesenchymal stem cells and all the stuff they need. So I'm sort of simplistic, and basically the growth factors are pretty simple: salt, sugar, and some vitamins and minerals. But the challenge really is protein, and as you've heard from Dr. Tressler, calf serum with mad cow disease probably is not the ideal source of stuff, so why not use stuff that is incredibly well screened, incredibly well controlled, collected in a GMP environment, platelets. So you can see this is just some basic science data from Mill Creek showing that 5% platelet lysate makes them grow faster than 2%, than 1%, et cetera. But it's also incredibly consistent because these are pooled products. And so you can see the doubling number per day, or measured in number of doublings over time, is incredibly consistent lot to lot. And I think it's really important from a manufacturing standpoint. You don't want one supplier. You want to have multiple suppliers out there. So it's good to have places like San Diego and Mayo and others developing these products, because God forbid the tornado hits, you want to have validated multiple sources, and maybe, one has better applications in growing one kind of cell versus another, and it's worth doing that basic science. So they actually have multiple phase 3 trials undergoing. They're supplying internationally. And so this is a byproduct, and the leaner we get and the better we get at not outdating platelets, the less that supply goes down. And so it is important to have multiple suppliers. And so what does one expect of a supplier? All of these things that you heard, I think, a ton about from the FDA speaker this morning. Who better to do that than those of us that have been being glared at by the FDA for the last 2 decades? We are certainly well consistent with that. So it's hardly only our own blood center, Oklahoma and Mayo, are also contributing to the Platelet Max product. And so in recognition of the time, I will end right there. Thank you.
I think that there's a lot of opportunities, and now just 3 days ago, China has just released their regulation on the stem cell clinical research and then the manufacturing standards. So I'm really looking forward for if there's any opportunity that both of the companies are looking for expansion in China, and then we would like to be partner of that, and we can do multi-center clinical trial on this, because we now have we are a hybrid bank for both the public bank and the family bank, and we are going to have around 8,000 to 10,000 samples each year. So we think we have a lot of resources, and then we want to explore collaborations. Yeah.
Clearly. The numbers are quite staggering that I've heard of in China, so I think you're in the right place, certainly for that. I don't know if there's any thoughts about that among the panelists. It was more of a comment than a question, but.
So I have had the honor of visiting many cities in China, and I've been really impressed at the progress in the both general blood banking as well as the donor screening there, and literally did visits 3 years apart when there was basically no nucleic acid screening to now widespread nucleic acid screening. So it's been amazing leaps of progress in general blood banking, and I'm sure that's spreading to cord blood banking as well with these new regulations.
Yeah, and on the cord blood expansion front, Angiocrine certainly realizes the potential of the market in China, and that is front and center for us. We have started having conversations already. No agreements inked, but you can certainly count on us paying attention down the road.
Clean rooms is they're very expensive, obviously, to maintain and have on site. So with all of the experience that blood centers have working, never using clean rooms and being able to provide sterile products to patients, where do you see kind of your manufacturing process fitting in a clean room or outside of one?
I'll deal with that. In fact, that was one of the thoughts that we had as to becoming more relevant to the rest of the world, and clearly that's an area that we excel at, is expanding these cells in a GMP environment. And so many of the biotechnology companies don't have the resources. It's extremely expensive, as you indicated, with the clean room environment. To investors don't have a sense of humor at all about spending their resources on bricks and mortar and building up facilities. Well, we already have it. And so I think the future is blood and tissue centers, particularly the more advanced blood and tissue centers, offering their sites as essentially contract manufacturing organizations for these companies where they can expand these cells. So does that address your issue or question? We can. Oh, absolutely we can. Absolutely. Oh, yes. Well, and hopefully there would be these closed systems. Yeah, these closed systems where it may not be necessary, like these closed expansion systems. Any thoughts? Yes.
Hi, this is Rob, hiding around the corner from you. Actually, we have a hybrid system. We have both ISO certification clean rooms as well as non-ISO certified processing rooms. And a lot of it's driven by, is the process we're using a closed system? If it's a closed system, you're not mandated to be in an ISO clean room. We have other systems that are hybrids. We call them semi-closed, which is a euphemism for they're open. Okay, hello, regulatory folks, we get it. And we do those in a clean room. We obviously, right up front when you're working with a potential customer or collaborator, you want to find out what their process is. And that's part of what I alluded to, know the resources, know the capability and the bandwidth of the blood bank you're working with. Do they have the ability to work either way? Because staying out of the clean room is a cost of goods effect for you guys. Staying in a clean room is a safety assurance effect for non-closed systems. So you want to have both, and depending on the manufacturing process or whatever system you're working with.
Brilliant. Okay, I believe we're out of time. Are we out of time? Yes. Okay. Thank you all for your attention, and it just so happens I'm going to put in just a plug for BioBridge Global and GenCure. We're looking for a senior scientist, so if you happen to know of a PhD, a cell biologist, a molecular biologist, developmental biologist who happens to have experience in these orthobiologics, wounds kind of stuff, let me know. We need to build up a lab. So thank you very much. Cheers.